He also wanted to look at differences in timing and the types of pesticides and see how that would affect bees. And the most significant drivers that were causing this relationship were actually insecticides that were applied after the flowers had finished blooming, and also fungicides that were applied before the flowers had even bloomed. And the strongest of these two were fungicides, stronger than insecticides. And so this is surprising for a variety of reasons. First of all, these insecticides or fungicides, they're not even being used when the flowers are blooming on the tree. It's before and after. So the bees are still visiting the trees during this time for some reason, or if there's pesticides that could be on any understory weeds that they're visiting, they're still becoming exposed to pesticides somehow. And fungicides is interesting because for a long time it was thought that this is benign for bees. Fungicides are designed to attack fungus, so why would they impact insects? But this study and others that I'm going to mention later are starting to show that certain fungicides can actually have negative impacts to bees. And fungicides are often applied during bloom and after bloom and before bloom, and we need to really rethink the times that we're using them. So the take-home message for the study is that pesticide use in 1 year can significantly impact bee fauna the next year.
So the good news is, coming out of all of this, that there is a way to buffer against these negative impacts of pesticides, and this is through natural habitat. So in addition to taking the samples and the surveys of the pesticides that were being used, they also looked at the percent of natural area that was surrounding these orchards. And what they found was as percent of natural area increased, both species richness and abundance increased in 2011 and in 2012. And this habitat didn't need to be present right in the orchard. It was within a 2-kilometer radius. So bees that are found in this orchard here, exposed to pesticides, may not feel the pressures as bad because they're surrounded by so much natural area, compared to bees that are found in this orchard with pesticides, found surrounded by a lot of agricultural area. Okay, so now let's shift the focus and look a little bit at what's going on with honeybees. So this data I'm going to share with you is also a very small sample size from a small study that was conducted by Paul Cappy. He's the state apiculturist for New York. And he collected this data for the National Honeybee Survey. And what he did was he collected pollen from 10 commercial beekeepers in 2011, 2012, and 2013. Then they looked at what pesticides were present in this pollen, and you're able to make this pie chart. So in these 30 colonies, over these past 3 years, there are 40 miticides present, 21 fungicides, and 26 insecticides, including neonicotinoids. It's interesting that the most prevalent pesticide that's being found in these colonies is miticides that the beekeepers are applying to try to control for varroa mites. But we're also seeing a lot of intake of fungicides and intake of insecticides. So this study is showing, yeah, there's pesticides present in the pollen, but it's not showing whether or not they're actually doing anything to harm the bees. And for that study, we began it last summer. It was led by Scott McCart, and we're still going to stay tuned for some of the results that he's having, but I'll introduce that study in a second. Okay, so the most common of these pesticides they're finding, the most common fungicide is Captain, the most common herbicide is atrazine, the most common neonics are thiacloprid and acetamiprid, and the most common insecticides, chlorpyrifos and cyhalothrin. I don't know if I pronounced those right, but chlorpyrifos is an organophosphate and cyhalothrin is a pyrethroid. Another interesting thing is we're seeing the same relationship with natural habitat in honeybees as we are with wild bees. So this plot was also made by Paul Cappy, who looked at the amount of colony losses beekeepers were experiencing in 2012 versus the time they're spending in agricultural land versus natural land. So the more time the colonies are spending in natural land, the lower their losses are, compared to colonies that are found in agricultural land for most of the year, which are experiencing higher losses. So again, that importance of having that forage available that is free of pesticides for bees, and for wild bees, it also provides safe nesting sites. So this ongoing study that's happening in New York to build upon this and to see how pesticides are actually impacting bee health is being done right now in New York. Scott McCart put 120 colonies among 30 orchards in New York. And that's no small feat trying to move that many colonies of angry bees that don't really want to be moved or touched. So every 2 weeks he went back and he sampled pollen and looked at the pesticide residues that are found within that pollen. And there's still a lot of analyses that are to be done to determine exactly what we found. So that will probably take a few more months. But in addition to pesticides, he's also looking at a lot of other factors affecting colony health. He's looking at pathogens and parasites, the types of crops that are surrounding these orchards and the percent of natural and agricultural land, and also the diet. What are these bees foraging on? How much of it is actually apple pollen? How much of it is from the natural forage around them? Okay, I'm going to take a second now to talk a little bit more about fungicides. And I'm sorry for how busy this slide is. I tried to summarize it all on one slide, but I'll take you through it so it should be pretty easy to understand. So in addition to Brian's study, we're also seeing that there are sublethal effects for bumblebees and honeybees in the form of impacts on foraging behavior. With honeybees, we're seeing that fungicides can actually alter the beneficial fungi that's found in this bee bread, or the food that they're feeding their larvae. And we're also seeing increased larval death. The most interesting thing to me is this concept of interaction effects. We're seeing fungicides interacting with a variety of pesticides to increase their toxicity. Things like miticides, pyrethroids, neonicotinoids, and we're also seeing an interaction with pests and pathogens, whereby certain fungicides can actually increase the susceptibility of some bees to develop fungal infections, which is really interesting, and I can talk about that with anyone after if they're interested. So just to show that interaction effect, I took this figure from Johnson et al.'s 2013 study in PLOS One, and it details this very nicely. So along the top, these are all miticides, and then along here, these are miticides, and these are different fungicides. So what they did was they exposed honeybees to these miticides, and they wrote down the LD50 values, the lethal dose 50, or the dose required to kill 50% of bees. And you can see that there's a lot of variation in here depending on the pesticide. For amitraz, it's probably the most toxic in this group. It only takes 3.66 micrograms in order to kill 50% of those bees. Thymol, which is more natural, it comes from thyme oil, you require a much higher dose in order to kill bees. So those are the LD50 values. Then these researchers first expose these bees to a miticide or a fungicide, and then expose them again to this miticide along the top. So when they're pre-treated with a fungicide or a different miticide, you can see that these LD50 values are changing. The more red it gets, the more toxic it becomes. So a really drastic example is if you pre-treat honeybees with prochloraz and then expose them to taufluvolinate, you can see that it only takes 0.01 microgram in order to kill bees, whereas before it took a much higher value. This is a simple lab test that was done by Cynthia Scott Dupree in Canada. There are a variety of other tests found out there in other literature. And what she did was she exposed 4 species of bees. We have alfalfa leafcutter bees, the common eastern bumblebee, the blue orchard bee, and honeybees. She exposed them to a few different pesticides. We have 2 neonicotinoids. Deltamethrin is a pyrethroid, and spinosad is a bacterial insecticide. And you can see that their lethal concentration values are very different. In general, the alfalfa leafcutter bees tend to be more sensitive than other species, but you can still see that there's so much variation out there. We can't just apply the toxicity of one species to encompass other species. So perhaps a good example is deltamethrin. Bumblebees and the blue orchard bee can handle much higher concentrations than can the alfalfa leaf-cutting bee. So to wrap this section up, I also want to remind you that pesticides are just one piece of the puzzle. There are all these other factors that I mentioned earlier, and these factors are not acting alone. There's a lot of interactions among them. Some of them I talked about today include fungicides interacting with other pesticides to increase their toxicity, some fungicides increasing the susceptibility of parasitic fungal infections, nutrition and loss of habitat can buffer against pesticide stress, and a lot of other management practices can also play into these other factors as well. Okay, so what can we do to help? There are 3 basic ways that we can actually help bees, and I think I have 2 minutes left, so I'll go through them rather quickly. We can use pesticides responsibly, practice integrated pest management, and a big one is providing pollinator habitat. So a few ways we can practice pesticides responsibly. Number one, I don't need to tell you guys this, but the most important thing is to follow label laws. There are very long inserts that come inside these pesticide brochures. For instance, imidacloprid is an 8-page information sheet with cautions and instructions on how to follow it properly. Also, these labels are changing. The EPA right now is trying to improve their label laws to protect pollinators. They're putting this bee advisory box on some of their pesticides, including neonics, to bring more attention to that. So every time, follow the label. Spray at night or in the evening when bees aren't foraging, and also when the air is calm to reduce drift into that natural forage that's also nearby. Avoid spraying during bloom. We already know this for insecticides, but maybe it's time to start considering this for other pesticides like fungicides. This can help reduce those acute bee losses, but we know now that it might not reduce the amount of exposure that bees are still coming in contact with. That's happening before flowers are blooming and after flowers are blooming as well. If you are a grower and you are renting honeybee colonies with beekeepers, having good communication with them, letting them know in advance, a few weeks in advance of when you're spraying so that they can come and cover their colonies or move their colonies if they'd like to. Choose a pesticide with lowest toxicity that will provide the needed pest control. If you're looking at multiple pesticides that have similar toxicities, look at the one with the lowest residual effect, the shortest period when that pesticide is actually effective. So there's a lot of databases and websites out there that show the toxicity of these pesticides to bees. One that I often use is called AgriTox, but even Wikipedia will post a lot of these toxicities. So do the research ahead of time. The formulation of pesticides you use can also impact bees. Choosing sprays and granular pesticides over dust and wettable powders. So dust and wettable powders have more capability to drift in the environment, and some bees have actually been seen collecting dust pesticides as if they would pollen. And then lastly, for using pesticides responsibly, reduce that planting dust that happens on treated seeds. Companies are getting good at improving their formulations, and some have actually made wax treatments that reduce that dust a lot, and having deflectors on your machinery to try to keep that dust down from drifting into the environment. I'll just go really quickly through this. Practicing integrated pest management. This is using multiple methods to control for pests and ultimately lowers the amount of pesticides that you're using. There's a variety of ways. Incorporating habitat that encourages natural enemies. So if you have aphids as an issue, try to have some habitat and ways that you can improve and increase the amount of aphid midges that are present. Planting disease and pest-resistant crops if you're comfortable doing that. There are some genetically modified cucumbers, for instance, that are resistant to powdery mildew, so you can use fewer fungicides. Also, crop rotations have been shown to reduce soil pests and reduce soil diseases. A very key part of integrated pest management is monitoring pest levels, actually going out there and seeing what your pest problems are, and if you actually need to be using pesticides at all that season. And again, there are alternate pest methods you can use, pheromone lures, more natural chemicals, etc. I am not an expert on integrated pest management, but this website, the New York State IPM website, has a lot of good information, and there's also a lot of speakers that are here today that know a lot about IPM that you can ask your questions to. And then my last slide is ensuring plenty of natural habitat in agroecosystems. I've demonstrated the benefit of having this habitat to buffer against pesticide loss, to buffer against honeybee colony losses. It provides safe nesting sites and forage that's free of pesticides. And right now, they're finally starting to come out with actual numerical recommendations. So this is for farmland, and right now the recommendations are to plant 1 to 4 hectares of forage and 1/2 to 2 hectares of nesting resources per 100 hectares of farmland. So that's very specific, but people have been asking for these specific recommendations for a while, so it's good that they're starting to come out. And there's more details in this publication here, Dix et al. 2015 in Ecological Entomology. So if you don't have a lot of that natural area within 2 kilometers of your orchard, there's a variety of ways that you can implement it that have been shown to benefit bees. Things like hedgerows, wildflower strips, adjacent fields, and field margins both increase the abundance and diversity of bees. And Heather Connolly, I mentioned her earlier, she's speaking in the afternoon, but she does a lot of work with wildflower strips and bees, and she'd be a great person to talk to. So with that, thank you for listening. If you want any more information about pollinators, you can email me at this address or visit our new website, www.pollinator.cals.cornell.edu.